Hey there, science enthusiasts. Ever wondered how to isolate DNA from lentils? Today I'm going to show you how to do just that using the CTAB method. Sounds fancy, right? But trust me, it's simpler than you think, so let's dive in. First things first, safety. Always wear gloves and safety goggles when handling chemicals. Safety first, people. All right, let's get started. We'll need some lentils, of course. About a handful should do. Rinse them with distilled water to remove any dirt or impurities. Once that's done, transfer them into a mortar. Now let's grind them up until you get a fine paste. This helps break down the cell walls. Next, we'll need our CTAB extraction buffer. CTAB stands for Cetyltrimethylammonium Bromide. It's a mouthful, I know, but it's essential for breaking down the cell membranes and isolating the DNA. Add about 10 milliliters of CTAB buffer to your ground-up lentils. Mix it well. Now heat the mixture. Place it in a water bath set to 65 degrees Celsius for about 30 minutes. This helps to further break down the cell walls and release the DNA. Make sure to stir it occasionally. After heating, let it cool down a bit. Next, we'll add chloroform isoamyl alcohol. This separates the proteins from the DNA. Add an equal amount to your mixture. Shake it gently for about 5 minutes. Remember, gentle is key here. Now it's time to centrifuge. If you don't have a centrifuge, you can use a makeshift one at home. Spin the mixture at high speed for about 10 minutes. This will separate the phases. The DNA will be in the aqueous top layer. Carefully transfer the top layer to a new tube. Add cold isopropanol to precipitate the DNA. You'll see it start to form a white stringy substance. That's your DNA. Let it sit in the freezer for about 30 minutes. After freezing, centrifuge the mixture again for 5 minutes. The DNA will form a pellet at the bottom. Pour off the liquid carefully and wash the pellet with cold ethanol. It's like giving your DNA a little bath. Finally, let the pellet dry for about 10 minutes and then resuspend it in a small amount of TE buffer. And there you have it. You've successfully isolated lentil DNA using the CTAB method. Remember, practice makes perfect, so don't get discouraged if it doesn't work perfectly the first time. Keep experimenting and learning. Thanks for watching and happy DNA isolating. Don't forget to like, subscribe, and hit that bell icon for more cool science videos. See you next time.